Hello, everyone, and welcome to another edition of the Edric Show. I am your host, Edric Jerome. This is the place for intelligent conversation with interesting people. Go ahead and hit that subscribe button, ring that notification bell, and you'll get notified when I post content each and every week. I've really been looking forward to this discussion today. My guest is science and technology journalist Fabrice Delaye. He is the author of the new book, The Medical Revolution of Messenger RNA, which tells the dramatic story of the development of messenger RNA and its global impact on the creation of life-saving vaccines. Fabrice is based in Switzerland, and he is an editor of the magazine Milan. He's now a reporter at large for Heidi.News in Geneva. Fabrice, welcome to the show.
Thank you. Hello, Hedrick, and hello to your audience.
Thank you. Thank you for joining us, sir. Very happy to have such a distinguished guest today. So let's get right into it. Such an amazing story. So tell me about how you first got interested in telling this important story about messenger RNA, and why did you decide to write this book?
So I'm following life sciences, and you can imagine with the pandemic, we were on the brink, writing every day about everything that was happening, lockdowns, or all of that. But in April 2020, the United States started with the warp speed operation to develop a vaccine as fast as possible, and I hear basically for the first time about vaccine based on messenger RNA. And then I followed the story during the year, the development. I get acquainted with BioNTech in Germany, with Moderna, who were the two companies developing this vaccine that has been selected by the US. And suddenly they pop up really fast. It was in July 2020 as the two potential leader, because they were entering into phase 3, the big clinical trial to prove the thing. In November, they get with the results. It was favorable. We were expecting for FDA to grant emergency authorization, which happened in December. And then you rewind then, and you are like, my God, it takes 10 months to develop that. And billions of people are going to be injected with such vaccine. Everybody was asking at that time, how is it possible it has take only that 10 months? Normally vaccine are developed over many, many years. And I say, well, that's a pretty legitimate question. And then I start to understand the roots of this technology, where it was coming from, why we did not have been hearing a lot about it before. Moderna was not a very famous company. Only few people know about it. BioNTech even more. And so I try to find the story behind, like for my own information. And I didn't find it. There was none. It has never been written. So I start to reconstitute, and soon I find myself into an incredible story with incredible characters, very strong personality, and also people that have been sided by the pharma industry, by the mainstream science, which was all about DNA, which is in slightly different molecules, even though there is relation between the two. And you know, broken careers, people that have been demoted in their university, and still they stick to it. So some of them stick to it, and they step by step develop this science, fundamental science to start with. That is the roots or the basis of the technology, which came naturally a little bit after. And so, yeah, I find myself with a pretty, I always say I try to write it a little bit as a thriller to make it easy, not too scientific, not too complex for people to understand. So it's very human oriented. The book is really about the people who did it. It's their story. And through their story, you understand the science. That's a little bit how it is working. And then I published that for my journal into various episodes. There was like 10 episodes. And soon I was contacted to make a book about it in French to start with, to transform those chapters, those articles into a book, and also in a documentaries, which I hope will be available in the US in some months. And then with Cold Spring Harbor Laboratory Press, who has been also always involved in this. Cold Spring Harbor laboratory researcher were involved in this long story, so they were interested in having not too much of a science book, but more like the story of the scientist. It's more the story of the scientist than the story of the science, if you want. But through people, you can say a lot of things and explain why they are hitting walls, how they go through the obstacle. So that's a little bit of the genesis of this project.
Before we get to some of the characters and the interesting people in the book, let me ask you if you can briefly, for folks, lay people like myself, what is messenger RNA and what is the medical significance of this development?
So I have to go back really at the roots of what is called molecular biology, which is certainly, together with information technology, has been the big driver of science for the last 60, 70 years. In 1953, the DNA structure has been discovered. So we know that in the cell, there is information that are basically coding for everything that is happening in the body. We have something like 37 trillion of cells in our body as human, and each of them has this code. But on the other side, at the other side of the equation, if you want, we have proteins, even more numerous. And those proteins are very, very diverse. There's hundreds of thousands of them that are different, doing, insulin that is allowing you to allowing your body to metabolize what you eat, erythropoietin, that is another protein that is transforming the oxygen that we breathe into something useful to oxidize basically into our blood cell, what we need to get energy, etc., etc. Hundreds of thousands of them. And so the question was, how do you go from the memory, the DNA, which is the big central computer, into the protein, which is like the software, which is the things that do things, or that would run a video, if you take the analogy. There is something in between. And this thing in between is messenger RNA. Sorry, it's a molecule that basically, there's an enzyme that is triggered by information cells receive. This is what I need. I need insulin to ingest what I've been eating. So it will go to the gene sequence of the insulin. It will just take this sequence. You don't need the rest of the DNA. You just need the sequence for the insulin. The messenger RNA coding for the insulin production will go into a little factory we have in our cell, deliver the plan. This factory will make the protein, and then the protein will go in the body and do what it has to do. It's everything. While I'm speaking, just the word I say is produced by certain neurons that are going to use this or that. I mean, it's a very, very central thing in the body. So it's DNA make RNA, RNA makes proteins. It's the mantra of molecular biology.
Let's talk about a couple of the notable people in the book who were recently awarded a Nobel Prize for their work. And this is such an amazing discovery, and it literally saved thousands, if not millions, of people's lives around the world. So this is not a small story. Yeah. Let me ask you now about the scientists, Caitlin Carrico and Drew Wiseman. Tell me about their importance, some of the struggles, and some of the things that you highlight in the book about their journey to ultimately receiving a Nobel Prize.
So first, Caitlin Carrico was interested in researching messenger RNA for many, many years. But from the 70s, from her study in the 70s. But she started in Hungary. And at some point in the beginning of the 80s, in Hungary, she was blocked in her research because first there was problem with, it was the end of the Soviet era and the economy was collapsing. So the research program she was in were not maintained. And also she has problem to access beyond the Iron Curtain to colleagues and to also produce that she would need for her research. So she decided to move in the United States in the mid 80s, and then she continued her research, but she hit a wall, literally, because she was. It was the golden age of DNA. And people was thinking that if you can control the DNA, which is the king on this chessboard, why would you need the messenger RNA that is just a pawn? It's just something that do, a little. It's the messenger. It's something that do something, but it's not that important because if you control the king, you are mastering what you want. It will disappoint later, but at that time, she has a hell of a time. She has been spending 2 years writing grants at her university. At that time, she has joined University of Pennsylvania, and she didn't get those grants. Her career was really, really into a lot of stress. At the same time, Drew Weissman, who started at the lab in Bethesda at the National Institute of Health as a researcher. It was this time, end of the 80s, beginning of the 90s, it was AIDS. Everybody wanted to find a way to develop a vaccine for this terrible disease. And Drew Weissman was one of the leading researchers in that field, and he was trying to develop a vaccine for AIDS. But it's extremely complex because AIDS is a very difficult to target virus because it mutates so much. It uses our body to mutate, our DNA to mutate. So it's constantly changing. It's very, very difficult. It would be like you have to basically make a vaccine per person differently each time. So it's very, very challenging. And so at the end of the 90s, they met. They were in the same department at UPenn, and Drew Weissman has just arrived. And they met, and Kati Kariko they were at the photocopy machine that they have their first encounter. And Drew Weissman didn't know exactly how to use the machine, what was the rule of the university. So she explained him, and then they start discussing. And she started bragging about her messenger RNA, that it would be awesome to do a vaccine with it. And then because it was in vaccine development, he said, why everything else is not working? So why not trying to find a way with that? Soon after, some years, when you say soon in science, generally it's 1, 2, 3, 4 years. But right. Anyway, they started to develop mRNA, but they discovered something that has been observed before, is that when they were using those messenger RNA to deliver this piece of information, they wanted to code a specific protein of a virus, they were obtaining inflammatory reactions that were not expected. And basically, this inflammatory reaction makes this candidate vaccine inefficient. They destroy it before it does its job. So they have been starting to look at what could have been the mechanism. And at the time they discover there is a protein. I'm not going to go into the detail there. Sure. To read in the book, but they basically discover a certain mechanism, and they discover a way to sort of go around this mechanism. And they publish it in 2005 in not a very prestigious journal. I think it was the Journal of Immunotherapy, which is a good journal, but it's not like Science or Nature. And they were expecting everybody would be calling them. Drew Weissman and told Cathy that they have to expect a lot of call when the article would have been published, and there was no call. Nobody was interested. And so they were pretty disappointed. They tried at the time to make UPenn take a patent on the invention. They tried to develop a company, but it didn't work. And they basically were lacking. So you have to imagine you have the messenger RNA, which is very fragile. It is something that is destroying the body within minutes. Normally, there is other proteins that are made to destroy. And so you need to protect it. And to protect it, you are using some kind of liposome, it's called, or now they call them lipid nanoparticles, which is basically grease that you put around the thing to protect it and to also allow it to better function in the body. And this technology was developed in Vancouver in Canada, but there was a big fight there between the inventors and companies and etc. And they were not able to put their hand onto this technology before the mid 2010, like in 2014, 2015. And so they get delayed a lot by that. But the companies that has been developing that in Canada, they find out their fight sort of quiet, and other were developing, and Moderna, and but there's still a lot of patent fights in this environment.
Sure, yes. I had a question about that coming up later too, by the way.
Yeah, sure. So that's the contribution, and if I can say, I've been meeting them on many occasions. You cannot imagine more different character than Cathy and Drew. Drew is the first time I spoke with him, it was unbelievable. It's the first person I know that when he's ending a phrase, you can literally hear the point at the end of the phrase. There's no more words. He has said what he has to do. While Cathy is incredibly outspoken, she's very empathic and very sympathetic and very passionate. So very different character, but complementary, I believe.
Going through the struggles they went through, when you heard that they had been awarded the Nobel Prize for their work, how did you feel? And did you reach out to them? Did you have conversations with them after they'd won?
Yes, with Cathy, not with Drew. But it was an amazing thing, because they have such a difficult time for so many years. People saying to them it's a dead end, you will never succeed, and etc. So this recognition is extreme. And Cathy says something that is very important, and I think Drew thinks exactly the same. They get the Nobel Prize. They have a major contribution that has been particularly useful for vaccine development, for prophylactic vaccine development. But mRNA can be used for cancer, for different. So there it's a little bit different technology, but they say it's a collective enterprise. There's so many people involved. I think for the book I've interviewed something like 60 people, and I could have interviewed 600 or maybe more. That has been at some point, a little contribution there. Might be a mistake, but when you have something it's a failure, well, at least you've closed that track. You know it's not the right direction, and other can build on that and say, okay, so why don't we try this now? And so I think it's important that she mentioned that there's a lot of people that has been involved in this research over time.
You mentioned patents earlier, and that leads me to a question I have about that, because typically in these types of advancements in science or discoveries, there tends to be fights over ownership of the patents, who owns it, who owns the technology, so forth and so on. Can you shed light on the current status of the patents surrounding messenger RNA, given some of the past legal challenges, and what are some of the issues around intellectual property particularly around applications in use for vaccine development?
Yeah, so well, first thing is some patents have a certain lifespan, typically 20 years. So everything that is older than 20 years is now in public domain. I was mentioning this publication by Cathy and Drew in 2005. It basically means that in 2025, in fact, in 2024, because the patent UPenn passed will be in public domain, everybody can use it. So it's the end of the patent fight. But yes, there is a lot of they mostly are around those lipid nanoparticles now, because the mRNA technology is quite established, and as how you synthesize mRNA, how you code them, all of that has been done over the last 30 years, 35 years. So it's okay. This is not so much of an issue. What is an issue is really the delivery mechanism, and there will be a lot of research that is still necessary to enlarge the potential of mRNA to other disease like cancer, like cardiovascular disease. The potential of mRNA is extremely wide because it's an information technology, basically. We are made of, our cells are made of proteins. So it's any proteins you can produce. So I was giving you the example of insulin. So remember that when insulin was produced, it's not produced inside the body. To produce insulin with modern biotechnology, you use bioreactors, and you code DNA into some organism that produce the insulin for you. You harvest it, and then you give it to people. But imagine now you can send something in an mRNA, sorry, and say, oh, mRNA, please produce this insulin for me. So insulin won't be maybe the best example because you need so much quantity, and also it's time sensitive. You don't need it all the time. You need it when you have been eating. So you will have something like a switch. So it's kind of a tricky thing to create that switch. But for people with diabetes, that would be absolutely extraordinary. But it's a difficult one for mRNAs, this one. While for cancer, there's no doubt, cancer is basically, we develop cancer all our life, as a matter of fact, but we don't know because our immune system takes care of it. When the immune system sees that there is cells that are starting to be abnormal, you just kill it. But the cancer has a way of hiding from the immune system at some point, and then it is when it develops. So if you are able to code for these proteins that are signaling to the immune system, hey, this one is abnormal, wake up and do something about it. I mean, I'm caricaturing, but it's basically what it is. Then you will be able to address cancer, and there's already a lot of research there. But in this case, you have a fight between CureVac, who started with cancer vaccine, and BioNTech. There is a trial that is going on because CureVac is saying to BioNTech, "Oh, you take things that were our intellectual property." That's life in this biotechnology pharmaceutical world. There's a lot of intellectual property fight.
Another question for you, if I can. Usually, scientific developments, things that get rolled out to the public, to the masses, at times there are ethical challenges or ethical concerns on behalf of either the consumer or behalf of whoever. What are some of the ethical concerns, if any, around messenger RNA, and do you think these will become further evident down the road, or do you think maybe some of these ethical challenges can be worked out?
I mean, I think there is two types of ethical issue there. One has to do with patient safety, and when it comes to mRNA in patient safety, what you would what some people will point is the risk of gene integration.
Hmm.
But in the case of mRNA, you are not using gene. It's not like a DNA therapy or gene therapy where you are sending information inside the nucleus. You never go in the nucleus with mRNA. You stay in the cell. And so the chance that there is integration, I cannot say they're nil, because unfortunately there is one mechanism which is basically something that is that the AIDS virus is HIV virus is very good at using. It's called reverse transcriptase. It's exceptional. The chances are super teeny, but you can never say 0, but they are extremely teeny. So on this side, the risks are, to me, the ethical question are very limited. Frankly, it's almost a non-issue. Then there is side effects. I tend to believe there is side effects with the vaccine. It cannot be stated otherwise. Like there is side effect with basically every drugs, every medical product. That's part of life. Certain people react differently than most of them. In this case, the ethical debate is about what they call risk-benefit balance. There is some risk, but if at the end there is much more risk of letting the virus free, you will have much more people with very bad, not only side effect, those people will die massively, 20 million in the case of COVID. So, it's a question. I know it's a very difficult question, because for the people who suffer the side effect, it's terrible. They are suffering something. Like in some cases, it has been associated with some cardio condition, not lasting ones, but still existing. And for those people, when you tell them, yeah, but you have saved other people, they say, well, yeah, but I'm suffering. So, they don't like it. Understandable. So that's where the ethical debate is with side effect. And finally, there is a big ethical debate. I think it's the most important one. It has to do with fair access, with equality. We in June 2020, the WHO, the World Health Organization, gathered a meeting in it was a Zoom meeting, but with all the head of state, from the world, or the minister of health, and everybody say, okay, we are going to do like this. It will be country independent. We don't care about who gets first in terms of country. What is important is that first we are protecting the people the most at risk, which mean frontline workers and elderly people with weak or immunodepressed people that have a weak immune system, that were the most people at risk. We didn't do that. When we got the vaccine, Americans say, we are vaccinating the American first. The French or the Swiss, or all the same. All the rich country vaccinated their population first. And the poor country, they get it much later. This is not something acceptable. And the ethical debate is not only because it's unfair to these people, but it's also because it's stupid. Because what you want is to stop the epidemic. And to stop the epidemic, you have to make the people the most at risk protected as fast as you can, because protecting them is protecting yourself. That's how it works in epidemic. It's different than other type of disease. In this case, you have, because the disease is infectious, the more virus there is, the most dangerous it is for everybody. So you want to stop it as soon as possible. So I hope that now there is initiative from WHO and others to have, or even Moderna and BioNTech are also doing things, for example, in Africa or Southeast Asia, for people to be able to access rapidly efficient vaccine. There is even they are even thinking of fast response, because mRNA vaccines are quite easy to produce. There is development that you can basically have sort of transportable vaccine manufacturing operation, and you will be able to send them where the epidemic is starting. So if you rethink of COVID, instead of vaccinating the planet, you would just vaccinate the area of Wuhan where it started, and it stopped there. You know, that would be the most efficient way, but it's still futuristic.
And political.
And political.
And political. And political. Yes, sir. Well, Fabrice, I want to thank you so much for taking time out of your busy schedule to talk to me here on the Edric Show. The book is titled The Medical Revolution of Messenger RNA by Fabrice Delaye. It is probably one of the most important stories currently on the planet, and I don't say that with any hyperbole. I think that's true, particularly moving forward, because these applications will have developments and impacts on humans down the road. So the story, you're almost like just starting the story. There's much more to say about this.
Yeah. Thank you very much, Edric.
You're very welcome. You're very welcome. This has been, oh, I'm sorry. If people want to get a copy of the book, more information about you, where can they go?
Oh, they can go on to the Cold Spring Harbor Laboratory Press website, where there is a link where they can order the book, and they can ask their librarian about it and order it. You can find it on Amazon, on Barnes and Nobles, and the classical way.
Sure.
Or on the Cold Spring Harbor Laboratory website.
Excellent. Fabrice Delaye, author of The Medical Revolution of Messenger RNA. Again, thank you so much for being on the Edric Show.
Thank you very much.
This has been another edition of the Edric Show. I am your host, Edric Jerome. As promised, this is the place for intelligent conversation with interesting people. Go ahead and hit that subscribe button, ring that notification bell. You'll get notified when I post content each and every week. I want to thank you for tuning in, and I will catch you on the next episode.